Hello, my name is Dr. Richard Harris, the host of the Strive for Great Health podcast, and welcome back to CBD Journal Club, where we dissect new research on CBD, one of our favorite holistic tools. I'm a clinical advisor and user of CBD Health Collection, who generously sponsored this video. Okay, let's dive in. The title of the article is CBD inhibits SARS-CoV-2 replication through induction of the host ER stress and innate immune responses. Basically, what this means is that CBD in this study was found to prevent the replication, meaning the increase in number of SARS-CoV-2 viral particles, by augmenting the innate immune response in host cells, basically human cells. So let's dive in. In the introduction, we all know SARS is a virus. It's been around for, well, this version of SARS for a couple years now. We've been dealing with this. It's a virus that has 4 key proteins. There's a spike protein, which everybody talks about. What's not talked about as much as some of these other proteins, the membrane protein, the envelope, and the nucleocapsid protein. SARS gains access to human cells by binding to the ACE2 receptor. And this is something that's been talked about. That spike protein binds to this particular receptor, and it causes the virus to be able to enter the cell, where it then hijacks our own host machinery, makes copies of itself, and then those copies bud out of the cell to go and infect new cells, or they'll leave the respiratory tract to go infect new people. After it enters the cell, the virus is translated into 2 large proteins to facilitate replication, which is making new virus, assembly, which is assembling all of those proteins into a nice, neat little package, and then budding, which is leaving the cell. CBD regulates the host responses, and it has some antiviral and anti-inflammatory properties. This has been seen in other studies. We know that hemp is a member of the cannabis plants and contains high amounts of CBD and other cannabinoids and less than 0.3% THC, while marijuana is cannabis sativa with more than 0.3% THC. We've covered this in some of our other videos about what's the difference between hemp and marijuana and the difference in CBD concentration and other cannabinoids and THC. Some studies report cannabinoids like CBD have antiviral activity against hepatitis C and other viruses. So this study wanted to look at, does CBD have the ability to inhibit infection of cells by SARS-CoV-2, or the virus that causes COVID-19. And in this study, they used human and monkey cell lines. They tested 4 independent preparations of CBD. They wanted a chemical version and natural, and they also tested related cannabinoids, so CBD, CBG, CBC. These are some of the cannabinoids that they looked at. They wanted to see, is it CBD or is it other cannabinoids? Which one has the effect? They also analyzed cell stress response. They looked at cell messengers for inflammation, cytokines. We've talked about those before. These are how cells talk to each other, especially related to things in regard to the immune system. And they also looked at interferons. Interferons are also messengers between cells regarding what's going on as far as the immune system, and they cause different responses depending on the different type of interferon it is. And then they wanted to look at direct antiviral activity. They also looked at the association of patients taking a prescription version of CBD for seizures with risk of testing positive for COVID. They validated the CBD using spectroscopy, which is the gold standard for looking at purity, how much CBD concentration. Then what they did was they incubated the cells with CBD for either 2 hours before infection or 2, 6, or 15 hours after infection to look at the differences. They used a CBD-infused hemp oil with 1,500 milligrams of cannabinoids in MCT, and they used a hemp extract from cannabis sativa with 54.7% CBD purity, 90 to 97%. So this is how they looked at the CBD alone versus the other cannabinoids. And when they used the hemp extract from cannabis sativa, they also isolated CBD, CBC, CBG, THC, and CBDV. So they were looking at multiple different cannabinoids. Now, they also did it in mice where they used 20 or 80 milligrams per kilogram twice daily injections for 7 days as a way to check in actual living vehicles. Mice. Okay, the results. The human lung cells were treated with 2 hours of CBD prior to infection. After 48 hours, they monitored expression of viral spike protein and viral titers. So viral titers is basically just saying how much of the virus is there in a certain amount of space. And then they looked at the spike protein as well. CBD was found to inhibit viral replication at non-toxic levels in human lung and monkey kidney cells. And that's very important. So there was no evidence of any toxicity or excess stress due to the CBD on the cells infected with the virus. And then it was found to inhibit the virus. They also checked the different variants. They checked alpha, beta, and delta, and the ability to infect was the same. So it didn't change based upon the different variant. And that's very important considering there's always going to be new variants, right? The virus is not going to stop evolving. We need therapies that are effective against multiple different types of variants. And we've talked about that before. We're actually going to talk about something on the podcast soon about a different response, not an antibody response, but about a T cell response. Next, they looked at other cannabinoids and found that only CBD had antiviral activity. In fact, combining CBD 1-to-1 with THC suppressed CBD's effectiveness. This is very important. So if you're taking CBD and THC, you may not get any antiviral effect based upon the results of this study. In this case, you want to go with something that's very high in CBD. CBD's main metabolite, which is called 7-OH-CBD, and CBD are both active and inhibiting viral replication, and both are non-toxic to the cells. Next, they looked at people taking prescription CBD for seizures. Now that dose is 1,500 milligrams daily. They looked at blood concentrations of the CBD and the 7-OH-CBD, and they found that based upon those dosing, that plasma concentrations, the amount in the blood, could inhibit viral replication. So we can't really extrapolate these results to lower doses, get the exact same results. But in theory, you would think that even at lower doses, there would be some effect, but it wouldn't be as dramatic of an effect. CBD is reported to decrease ACE2 expression in epithelial cells. However, this study did not show a difference. They looked at that as one possible mechanism because if it decreased ACE2 on the cells, then there's going to be less for the virus to bind to enter the cells to cause infection. However, CBD was very effective, about 95 to 99%, in inhibiting spike protein expression in host cells 2 and 6 hours post-infection, even in the presence of antibodies to the spike protein. So this is showing that it's working in a different mechanism than either our immune system or vaccine-generated immunity. CBD was partially effective, about 60%, at inhibiting virus 15 hours after infection. And it didn't seem to do so via processing or assembly, so it may do so via alternating other host cell processes. So they wanted to look at what was the possible mechanism. Cells treated with CBD for 24 hours show a suppression of SARS-induced changes in gene expression, including the expression of multiple proteins, the spike, membrane, envelope, and nucleocapsid proteins. What this means is that when the virus hijacks our own machinery, because it hijacks our own cellular machinery to make more copies of itself, when combined with CBD, there was no change in gene expression to make more of the spike protein, the membrane, envelope, and nucleocapsid proteins. They found that CBD, the viral cells had a pattern of gene expression similar to CBD alone. So what this basically means is that when the CBD was there with the virus, it inhibited the virus from being able to do viral things, and it made the cell act like the cell would just had CBD alone. Overall, these results suggest that CBD acts to prevent viral protein translation and associated changes in the cell. CBD was found to increase endoplasmic reticulum stress response. Endoplasmic reticulum is part of our cells. It's also called the ER. And what the endoplasmic reticulum is for is when proteins get made in our cells, they go to the ER to get folded into the correct 3D configuration. This is not something that we've talked about on the podcast before, I think. We've talked about how receptors have a 3D configuration and you need the right pattern to bind to the receptor, and then different configurations will bind differently and cause a different effect. One may bind and cause 100% effect, one may bind and cause a 20% effect. But this is due to that 3D structure, and that 3D structure is generated in the endoplasmic reticulum. So what happened is CBD increases endoplasmic reticulum stress response, interferon induction, and an unfolded protein response. And so genes associated with oxidative stress response were also induced. What this basically means is it's causing the cell to induce more of a immune type of response. ER stress typically happens when the workload on the protein folding machinery exceeds capacity. And what happens is this triggers a signaling called UPR, which increases three proteins, IRE1 alpha, PERK, and ATF6, to increase protein folding. So basically, when a lot of protein gets delivered to that endoplasmic reticulum to fold, it causes a stress response, which increases these certain mediators to increase protein folding. So it's like if you have a bunch of orders in your factory coming through, you increase the capacity by adding more workers or adding more machines or something like that. When you get the signal that there's more upstream, you need to make more product. However, prolonged stress, and we've talked about this on the podcast before, some stress, good, increases our capacity, prolonged stress, bad. This signal cannot be fixed, and UPR will trigger cell death. CBD increased expression of some of these proteins. What happened is CBD activates IRE1 alpha, but not SARS or PERK, but not PERK or ATF6. And then what happens is SARS activates PERK, and in mice, if you knock out IRE1 alpha, you get a reduced dose response to CBD. And so what this is signaling is CBD was found to induce interferon signaling pathway, which are messengers that are part of the innate immune response. SARS suppresses interferon signaling. So what SARS is doing is it's signaling this whole pathway to increase protein folding, but then it's not mediating it, which is leading to unregulated inflammation and cell death. CBD increased several important genes in the interferon pathway alone, but it was highly induced in the presence of the virus. And we've talked about this with CBD before. CBD regulates a lot of our natural pathways, and it works more under periods of stress. We talked about that with pain signaling, that CBD is more effective the higher your baseline degree of pain is. CBD also reversed viral induction of other cytokines associated with cytokine storm. Cytokine storm is something we've talked about with a robust immune response. And what happens is the immune system can go too far and damage cells. And this happens due to certain chemical messengers get released too much. The body overreacts to some type of pathogen and damages host tissue. Next, they looked at whether CBD can inhibit replication in mice. CBD was found to significantly inhibit viral replication in the lungs and in the nose 5 days post-infection in a dose-dependent manner. The low dose, there was a 4.8-fold decrease in the lungs, 2.7 in the nose. In the higher dose, there was a 40-fold decrease in the lungs and 4.8-fold decrease in the nose. And the mice showed no signs of clinical disease. Now, they're using much higher doses in the mice than what you would look at in people. Finally, they looked at medication records for patients on prescription CBD, and the patients on that prescription CBD near the date of their COVID test showed a 6.2% infection rate versus 8.9% in non-CBD controls, for a decrease of about 35%, or an odds ratio of 0.65. And the demographics were similar in both groups. Even more striking, in a subset of patients who are taking CBD on dates prior to the first COVID test, it was 4.9% versus 9%. So 4.9 in the CBD group, 9% in the other group, for a reduction of 52%, or an odds ratio of 0.48. That's pretty significant. So in the discussion, the results indicate that CBD and its major metabolite block SARS infection at early and even later stages of infection. It's much better early than later. Also, CBD reduced viral titers in the lungs and nasal passages of mice. That's very important because we know high titers are associated with more transmissibility. It's easier to transmit when the titers are higher because you'll put more of the viral particles out when you breathe or cough. And higher viral titers are also associated with worse disease. In other diseases, haven't seen any papers yet for SARS-CoV-2 in this regard, but we assume that it is the same, that higher viral load is associated with worse disease. Analysis of national sample of patients using CBD consumption at home to COVID tests showed fewer positive results. CBD induces interferon pathways both directly and indirectly, but it reduces cytokines associated with cytokine storm. This basically means that CBD was helping the immune system fight off the virus, but not going too far. The authors hypothesized that since CBD blocks replication after entry, it will be effective against mutants that have a different spike protein changes, because that's mainly what we're seeing, especially related to the vaccines, is that the vaccines and some of the monoclonal antibodies are losing effectiveness because of mutations in the spike protein. Now, some caveats here is, of course, CBD differs in doses and purity. We've covered that before, how you make sure you're getting a high quality product. Oral administration can cause lower amounts in the blood than as they were using injections and directly applying it to cells. So, and they were using much higher doses than what people conventionally take. So you can't just take the results of the study and apply it one-to-one saying, oh, I'm taking 20 milligrams of CBD, it's going to give me the same protection. No, it's not. And this was not a human trial. This was not a randomized controlled placebo trial. So we cannot imply causation in this trial. But it is a really cool mechanistic trial and showing that, hey, there's enough here that we should look at, and this is what the authors say, we should look at placebo-controlled studies of known concentrations of CBD to impact viral replication and disease course in viral infections like COVID-19. Well, I hope this was educational. Again, CBD is one of my favorite holistic molecules, and there has been studies looking at the antiviral and antimicrobial properties of CBD, and I'm sure that more and more research is going to come out as we look for safe, effective therapies for modern infectious diseases. All right, this has been Dr. Richard Harris on CBD Journal Club. Thank you for listening, and have a blessed day.